second service.
It's not funny. You already know you should.
Yeah. Go for it.
All right, let's do this. All right, so just a quick introduction to the beginning of the talk. My name is Mike Catarina. I'm a professor at Clemson in the Department of Plant and Environmental Sciences. And tonight I'm going to talk a little bit about some research that I've been doing in my lab group on insects in the region. And I'm going to talk a little bit about insects themselves. So, I do want to keep this kind of informal. If people have questions as I'm going, please feel free to raise your hand. I don't want you to forget them while you're waiting for me to finish. You might have to get up and get a beer or something like that and forget it. So feel free to raise your hand and we'll deal with it right then and there. I'm going to give you a little bit of background on insects in general before I start, as well as a little bit of background on myself. So that's where we're going to start. So I've been interested in insects for a long time. I can't say that it's like always been my life's dream to study insects, but you know, I was into them as a kid. I thought they were pretty cool. But it really wasn't until I was in college at Ole Miss where I took my first entomology class, and early on, my professor put one of these tiger beetles up under the microscope, and I took a look at it, and I was just smitten. It was like instant love at first sight. So yeah, this is a tiger beetle. The beauty, the intensity, intricate detail, and really, I haven't looked back. This has just been with me always. Hold it closer to my mouth. Okay. Sorry, I'm not used to talking with a microphone. We'll get it figured out. So within just a month or so, I was applying to graduate schools, and I was ready to make bugs my life's work. So it's really worked out. So before I get into the research, though, I want to talk a little bit more about insects in general. And I want everybody to know just how important insects are. There's kind of this perception that they're very small, and so they must be kind of insignificant. They're out there doing their thing, but, you know, whatever, they're not that important. Really nothing could be further from the truth. So, first of all, I mean, I'll admit there are some bugs that we don't like. You know, a lot of people are just scared of them, but there's lots of bugs that they bite us, they sting us. Things like mosquitoes can transmit diseases. You know, they'll eat our homes. You got maybe cockroaches scurrying around in your kitchen. I don't know what that's like. And then some of them will even do the unthinkable, eating barley and hops, our two favorite crops. So yes, there are a lot of reasons not to like bugs. But all of this is vastly outweighed by the good things that they bring. And really the first thing on the list that I would mention is pollination. So bees, of course, but also beetles and flies and moths are really important pollinators of all kinds of plants. In fact, pollination and the fruits and nuts that are produced by pollination are worth over $200 billion a year in crops globally. And to put this maybe in more concrete terms, pollination by insects is responsible for about a third of the food that people eat all over the planet. So, you know, there's about 9 billion-ish people on the planet now. If it weren't for insects, 3 billion of those people would be starving right now. So I think that's important. Another thing that insects do is there's a lot of kind of bug-on-bug violence out there. A lot of insects eat other insects, and we really can thank a lot of these predators and parasites for there not being more pesky insects on plants or buzzing in our ears and things like that. So up in the top left, we've got a dragonfly nymph eating a mosquito. Anybody here like mosquitoes? No, of course not. Thank you, dragonflies. We've got a caterpillar hunter down here eating a larva. We've got those little green things on that caterpillar are the larvae of parasitic wasps eating it. The wasps will parasitize the eggs of things. We've got ladybugs that everybody loves because they eat aphids. So these guys are doing a lot of natural pest control for us. And even in a lot of crop situations now, we will use things like parasites and predators instead of pesticides for a lot of things. So they're very valuable there. Anybody like fishing or birding? Lots and lots of fish and birds and even lizards, if anybody likes lizards, live on insects. They have to eat them. So all these other things in the world that we do like depend on there being a healthy insect population. So that's important. And then insects are very important recyclers in nature. Things like dung and fallen trees and dead animals and the leaves that are out falling in your yard. All these things are food to insects. And what they do is they'll eat these things, they'll eat the poop and then they'll poop it back out. And all of that gets turned back into soil. So that's a really critical service that they perform in the ecosystem of just recycling all this dead organic material. And then our forests and prairies can flourish. So all that's really, really important. So the good, bad, and the ugly. All right. I can't fulfill the trio there. There really are no ugly insects. They're all incredibly beautiful, and even just appreciating the beauty is a pretty important industry now. People go all over the world to see butterflies. There are dragonfly field guides. Even around here, there are people that will go out to see fireflies in Congaree or in the Smokies. So, there's kind of this ecotourism industry that's sprung up around the beautiful insects as well. So, there is a ton of value out there, and I think the lower left, well, this is a good friend of mine. That is a beetle called Heterius californicus. It lives in ant nests. So that one was in California, but we have these here as well, and they're actually guests in the nests of ants. So a lot of those little hair tufts are actually where they produce chemicals that smell good to the ants, and so the ants say, we like those guys, we'll keep them around. Yeah, so, I mean, the beauty there is maybe a little bit more in the eye of the beholder, me being the beholder, but they do eat ants. Yeah, they're parasites in the colony, but the ants don't. It's okay. All right, so with that, I'm going to move on to some of the research work that we've been doing. For the last several years, we've been really interested in my lab in looking at the diversity of insects that live in leaf litter. And so, this is a typical scene now with all the leaves falling off the trees. And, that's not just junk to be raked and burned and thrown away. That's a critical resource for a huge community of insects that eat it, that eat each other while others are eating it. And so we wanted to get to know this community better. Most of these insects are extremely tiny, 1 to 2 millimeters. You barely see them if they're just walking across the table in front of you, but very critical in processing leaf litter. And the place that we chose to do this, sorry, this question? What's that? Oh, so that's a pseudo-scorpion. So they're related to true scorpions, but they don't have the stinging tail. Very, very small, 2-3 millimeters, but very abundant. You can get hundreds of those in leaf litter. Yeah, I mean, I know after the talk, you guys are all going to go out and start looking through your leaf litter and say, I want to find that little thing there. That looks cool. But yeah, all this stuff is out there doing its thing without most people being aware of them at all. All right, so I really want to focus on some work that we've done over the past several years in the highest elevations of the southern Appalachians. So I don't know how many of you have been up to Clemson's Dome in the Smokies. So as you get up higher, hit about 5,000 feet, you see that the forest kind of changes from the sort of deciduous forest that we're used to around here to this conifer forest of spruce and fir. It's a really distinctive type of forest, and there's a lot of distinctive things that live there. So it's much cooler and moister than the widespread forests, and it's like this series of islands. So these are all the patches of spruce fir that we have in the southern Appalachians. From southwestern Virginia up there, number 1, around through the Black Mountains near Asheville, and out to the Smokies in the west there. And so these are little islands of this cool, moist habitat. And these trees were basically stranded there when this kind of widespread conifer forest slowly started migrating northward at the end of the Pleistocene. And the glaciers were receding. This cool coniferous forest was also receding. Some of it receded up the mountains where it was cooler and cooler, and then got stranded. So you've got all these little islands of habitat, and where you have this kind of isolation, this is where evolution really starts to kick in. Because you've got populations of things that can't mate with each other anymore, and they start to diverge. So this is an area of pretty interesting diversity. So in creatures that people pay more attention to, like salamanders, the southern Appalachians are a global hotspot. There's more species of salamanders in the southern Appalachians than anywhere else on Earth. And these are a couple of endemic species only found in these spruce fir patches in the southern Appalachians. It's also interesting in that we have some species that live in these spruce fir patches that mostly only occur in the north, even up into Canada. But it's cool enough at these high elevations that they can persist at the tops of these mountains. So things like the northern flying squirrel, the redback foal, these are like the southern outposts where those species can live. Among arthropods, there's been a lot less work done, but this is one species that's received some attention. So the spruce fir moss spider, this is like a baby tarantula in a way. It's a really tiny spider related to tarantulas. And all of their populations live only in these spruce fir forests. So this thing had become rare enough that it was federally listed as endangered a few years back, and people are monitoring that pretty closely. But apart from that, there's been really very little work done on the arthropods that live in the spruce fir forest. So this is what we want to do. So we got a National Science Foundation grant in about 2019 to go up and do some surveys. So just a real quick overview of the kind of work that we did to do this. We would go up into the forest, and my colleague Ernesto there is holding a sifter, and this is a sifter. There's a screen inside there. We put leaf litter and needle debris in the top of that sifter and shake it around, and the little things, which is mostly bugs and some fine debris, fall through to the bottom. We bag that, bring it back to the lab, and we put it in these Berlese funnels on the bottom left there. So there's a screen there, we put the litter on top, and the light dries the litter out, warms it up, and the bugs don't like that. They like to be cool and moist, so they try to burrow through and then fall into a little dish at the bottom, and then you end up with this just wonderful bug soup. And I mean, I can't tell you, every time we go out in the field and come back and load up the Berlese funnels, the next morning I'm just like, I can't wait to go into the lab and see what we've got. I mean, it's like every dish is a Christmas present. And then we can sort through those things and try to identify what we can. But there's a lot of that stuff that we can't identify. There's either no literature, no books that we can find that will tell us how to identify some of those things. So we wanted to do some DNA sequencing to help us identify them. But also to start to look at, how much evolutionary divergence there might have been. So are we seeing genetic differences between populations on some of these different mountaintops? And so Ernesto's loading up our little MinION sequencer there and generating lots of sequences. One of the things that we're expecting to find were cryptic species. So we're finding more and more that there are species that we really can't tell apart just by looking at them. Like Sasquatch. You knew where I was going. Sorry. That's all right. Yeah, no, I'm not talking about cryptids. Although, I'm always on the lookout for Mothman. So far, there have been no sightings, but we're keeping our eyes open. The cryptic species that we're talking about are these. And this is becoming more and more kind of a common thing in entomology, where almost every month somebody's publishing a paper saying, this thing that we thought was one species, well, actually there's 8 of them, or there's 16 of them. So the one on the right there, this is a skipper butterfly from Central America. And for a long time it was thought to be a single species. The caterpillars that you can see have a lot of color variation, and they fed on a lot of different kinds of plants, but everybody thought this was just variation. But using DNA barcoding and sequencing individuals from around all these different kinds, it became clear very quickly that there were actually, in this case, 8 species masquerading as one. These parasitic wasps on the left, similar situation. We thought there was one species that parasitized a lot of different host insects. In fact, it was 16 species, each of which was very specialized on a particular host. So this is the kind of thing that we're looking for. Maybe that there would be cryptic species on each of these different mountaintops. And to make a long story short, that is more or less what we found. So these communities are extremely diverse. In each of those little white boxes, the number on the top is the number of different arthropod species that we found on that mountaintop. So 100 or more or less on most of the mountaintops. And then the lower number is how many were only found on that one mountain. Yes?
Do you find that, like, in conifer forests and deciduous forests, that because it's like type of leaf litter, whether it's like mostly pine or like cypress, where you find like there's differences in diversity and like the types of species and the type of species? Is there a difference in that?
Yeah, so the question was, do different types of leaf litter, whether it's from like oaks or from these conifers, support different kinds of arthropods? And the answer is absolutely yes. Not 100%. I mean, there are some that are kind of generalist and can eat whatever, or they're actually eating fungi that are growing on those leaves, so they're less dependent on exactly what those leaves are. But for the most part, yeah, there's different types of litter, you're going to have different kinds of arthropods, for sure. Yeah. So the bottom number is where things really got interesting. So that's the number of species at each of those sites, each of those peaks, that were only found on that one peak. So around a third of the species on each mountaintop could only be found on that mountaintop. And this gives a really different picture if you want to try to conserve these sorts of communities. You're not looking anymore at, widespread species where, if we protect them in the Smokies, then, they've got a pretty secure home. Instead, the ones that are in the Smokies are actually different from the ones that are in the Great Balsams, different from the ones that are in the Black Mountains. It makes the challenge a lot more complicated, but that's reality. We really need to be thinking about these mountains quite differently. There's a lot more diversity than meets the eye. So that's really, the overall results of this project. But I wanted to introduce you to some of the key players and a few of the things that we found really interesting. So this is one of these that turned into a cryptic species complex. So Dasyceras carolensis, this thing that you see, had been described from a little bit east of Asheville over 100 years ago. It was about 1880. And, over the years, people found it more and more widespread up in southern Virginia, down into southern Georgia and Alabama. But it was all considered to be one thing. As recently as 1996, some people had looked at it. But nobody had done any of this kind of DNA work on it. And when we did, and we had over 100 individuals from almost this entire range. And what you can see in this phylogeny or evolutionary tree is lots of very distinct clusters. So the genetic divergence among all of these different places was very high. And so it looked very clearly like we were dealing with cryptic species there. Once we saw that, we went and looked a little bit closer at morphology. And I'm sure there's like one person in this room who knows what they're looking at right now. Does anybody else want to guess? Yeah. So this is basically the penis of the male beetles. And they kind of look still very similar, but there are really some important differences here. So along the middle part here, you can see that there's some little teeth or spines, and these are basically like little teeth that they get everted, like spines on a balloon, if you can imagine that. And if you look at those are really quite distinct among all these different species. And those have to interact with the female internal parts very particularly in order for her to accept them as a mate. So when you see that in insects, then you know you're dealing with different species. And these lined up perfectly with the genetic clusters that we saw. So with that, we felt pretty comfortable describing, in this case, 10 new species of these from throughout the range. Yes, sir?
How do you do like the DNA barcoding?
Okay, so the DNA barcoding, we take each specimen and we bathe it in a little chemical bath that isolates the DNA and clears out all the other things that aren't DNA. And then we do a reaction that pulls out just one little piece of the DNA that we can then compare among all the different species. And we can visualize that using that sequencer that you saw Ernesto loading. So we can see each little peak is an A, a G, a C, a T, and we can just read it off the sequencer. In a nutshell. Yes?
For the DNA samples, were they from the 1996 paper, their samples, or was it from what you had?
We didn't have any that they had looked at, but we did have some museum specimens. We were able to get DNA from some older things, but mostly that was all new things that we collected. The reliability is so much higher when you're using fresh material that I always try to get fresh stuff, but yeah, we can usually get this out of museum specimens too. Yeah. Yeah.
Do you use electrophoresis for the barcoding or?
No, it's not really electrophoresis anymore. That sequencer uses what are called pores. So there's like a membrane, and the DNA goes through the membrane pore, and there's a little sensor that basically detects the charge of what's going through. So the DNA is moving like it would in electrophoresis, but the detector is different. Yeah. Yeah.
Sorry, one more. What's up with the yellow triangle in Kentucky? What's up with that species?
Yeah, so that one is, that's actually not one of these. That's a different species that does look different. So it's not part of this cryptic complex, but that was quite an outlier geographically. We've only seen specimens of that. We haven't been able to sequence it. So we suspect that's going to be different too. But yeah, there's a few of these that don't quite fit the pattern. There's some that are more widespread. But in some of those cases, we haven't sequenced from throughout the range, so we'll have to wait and see.
So you think that's just a sampling gap then there?
I don't know. I mean, there's a lot of lowland there before you get to the Cumberland Plateau. And often the things on the plateau are really very different from things in the Southern Appalachians proper. So that was really outside our sampling range, but I want to do more for sure. Yes, sir. So we chose to mostly use Cherokee names for these. So those are mostly based on place names near where the specimens were found. Yeah, we tried to find names that were sort of. Yeah, they're so like Catalucci is like probably the original, more phonetic way to say Catalucci. So that's one of the mountains in the Smokies. But, yeah, I mean it's typical. We will often try to name a species for the place that it's from. Yeah, dissecting tiny beetles, that's a whole other topic. Yeah, you don't drink too much coffee in the morning. You make sure your tools are really sharp. We do it all under the microscope. Yeah. All right, anything else? All right. This one, we'll go through a little bit more quickly, but this is another genus that another student of mine out of university worked on, and it's really the same story. There had been one species in this group called Arthropods carolini, and we collected some things that actually looked pretty different. Like that pale one is totally blind. That looked like it was clearly going to be different, and the DNA supported that 100%. So, and the genitalia morphology does as well, really different things. An interesting aspect of this one was that we collected the immatures, the larvae of almost all of these, And if you think that the adults would be hard to tell apart, the larvae are like on a whole nother level. Nobody wants to look at larvae. But now we know what the baby beetles look like for almost all of these too. And we were able to match those up with the DNA barcodes. So that was a really nice study. This is in a group that was only known from Europe and Asia previously. So this doesn't really fall in the cryptic species category, but rather there are things that are just completely different that nobody's collected before or taken the time to look at. So these have got some really interesting kind of sawtooth ridges on the male antennae. And when I saw that, I knew there's nothing in the local fauna that would have those. And it took a little detective work to figure out that they're related to these European things. And now there's 13 species of these, where previously we had 0. And then some other groups that we haven't looked at in any kind of detail, but I just wanted to make you aware that they existed. Springtails are a group of really, really tiny arthropods. If you do take time to go out and look at the leaf litter, you move it, and you'll see these little white dots kind of bouncing in all directions. And those are springtails. But when you see them up close, they're gorgeous animals. They're really attractive, really diverse, and we've taken just a very shallow look at the diversity of these with a couple of undergraduates and students. But there's a ton of work to do here. Mites. Nobody looks at mites. The diversity is phenomenal, even tinier, but incredibly diverse and rich. Really important for the ecology of these communities and really need to be looked at. No, none of those are bedbugs. Bedbugs are true insects. They're related to stink bugs, but yeah, they happen to be blood-sucking. No, some of them eat plants, some of them eat fungi, some of them eat each other. They do all kinds of different things. And I'd love to have somebody specialize on these. That's like another lifetime or two. Okay, so basically to wrap up, the Southern Appalachians have an incredible insect fauna. The leaf litter is really rich, and I think a lot of these species are probably very important to maintaining these forests that do support other things we care about. There's lots of new species. Some of them are quite cryptic, but some of them are not so cryptic. It's really a matter of taking the time to look. Yes, sir? So the cryptic species are ones that look just like another species. You can't tell them apart by looking at them, but in fact, they're different species. They may smell different, they may have different DNA, but basically they've been mistakenly lumped together. Exactly. Yeah. Yeah.
For those samples from Appalachia, which had like the number of species, how large were those samples? How much litter did you take that you got that litter?
We try to visit every site twice. Ideally spring and fall, because it can be a little bit different. And then each time we go, we take about 10 samples, which doesn't mean much, but each sample is kind of like a gallon of litter. So it was a lot, but I'm sure those numbers would keep creeping up if we were to go back over and over again. Yeah, so how do you tell natural variability from species level variability? There's no clear-cut answer, but you ideally have a lot of specimens and you can see patterns. Like most of these individuals are bigger, or they've got more hairs on the head than these ones do. So it really does take repetition to be able to see those kinds of things start to jump out. Yeah, and then you just get to know, a lot of beetles have sexual dimorphisms, like the males, those antennae look different, and you more often see differences there than in other characteristics, so you kind of learn what to look for in different groups. Yeah. All right. Yeah.
How long did it take?
Yeah, so I have mentioned all these projects that I've talked about have been collaborations with students or student thesis projects. So the Dasyceras and the Lithobium were PhD students, so they spent about 5 years on those projects. The springtail papers I did with a couple of undergraduates in a year. So they did like a senior thesis with me, and we were able to do some work in that time. But yeah, it varies. Yeah, so the functions of the ridges on those antennae, I can't say. It's really hard to know. There are some beetles related to those where the antennae are enlarged, and they're really obviously like for grabbing the female. So it's a courtship thing. Here, that's not so obvious. I don't know. It's probably similar, but yeah. So many of these things nobody's ever seen alive. You know, you can't watch a dead thing behave. That's a problem. Yeah.
You got some names in them?
Oh yeah, all those names that I've shown are things that we came up with, and that's a whole other topic. Like, how do you come up with the names? You want to come up with something that sort of like honors the beetle and the area.
Do you have anything after you?
Yes. But I didn't do that. You're not allowed to name them after yourself. But in this really obscure corner of beetleology, I've collected some things that other people have then gone on to describe. Like, that's no real. That's forever. Well, so that detective work, I looked in the literature to see if there was anything similar to those beetles. I found publications from Europe that describe them. Oh, no. They've always been there. Just nobody knew. So the ancestors of those, so the ancestors of those came over, 10 million years ago or something. Yeah, exactly. Anyway. All right. So I think you all have time to read this by now. But it's really, there's a rich fauna that we need to understand better if we want to protect it. And these first fir forests are really susceptible to the effects of climate change. The stuff that's there is there because it's cool, because it's moist. Warming and drying are two of the things we most predict happen. So, I don't know what the prospects are of preventing that, or maybe moving some of these things a little bit northward to try to keep them safe. I don't know. I mean, that's a whole other topic. But we at least need to know what's there before we can start to think about solutions for conservation. And then really the bottom line is that there's a ton of stuff still out there to discover. And when I was a student looking at that tiger beetle, I had no idea. There's this sense that people have been doing science for hundreds of years, and all this kind of basic discovery is done. And for insects, we think 90% of insects are still unknown, undescribed. A lot of that's in the tropics, but a lot of it is here too. So there's a lot of opportunities for people to still come up and make really basic and exciting discoveries. So with that, don't forget, insects are both amazing and very important, and please spread the word. And just quickly, I want to thank a bunch of folks, students and colleagues who worked with me on this project. And a lot of funders, so we had really good cooperation from the state and national parks. As I mentioned, this was funded in part by the National Science Foundation. They're having some rough times these days, so they do good work. And then I also want to give a shout out to John and Suzanne Morse. John was a professor in my department who helped to fund my position and really made a lot of this possible. So with that, I will stop talking and be happy to answer all your questions. Thank you very much. Yes, sir. Three questions, I'm ready.
Unresolved, you didn't cover this.
Okay, good.
First one is the Mothman actually half and half moth.
Yeah, so at some point, a moth and a human became curious, and they loved each other very much, and the rest of the story.
Two, is there any commonality in DNA between insects and humans?
I mean, if you were to look in detail, you would find a lot of things in common. So the gene that we were sequencing is part of the mitochondrial DNA. Humans also have mitochondrial DNA, and the sequences are 70% similar. It's hard to put those kind of numbers on the whole genome, but yeah, we share a lot, for sure.
Third question.
Yeah.
It might be boring.
Yeah, we got all night.
Explain to me how a worm decides, I'm gonna crawl around for a while, then I'm gonna cover myself in stuff, I'm gonna dissolve into goo, and then I'm gonna come out with something else, like a fly.
How does a larva become an adult?
How does that possibly evolve?
I'm actually teaching a whole class on that this semester. So the answer is going to take about 12 weeks. The part of the answer is that while these insects are larvae, the adult is slowly forming inside. So there are little pockets of tissue inside the larva that are becoming adult-like. So the wings, there's little cell clusters that are becoming small wings. So, you know the pupa? So what's the chrysalis? When that comes out of the larva, at the end of larval development, all those adult structures are already there. So they've been ready. And so after that stage, it just has to inflate them, basically. They're all there in miniature. So it's a longer process than just magically the adult comes out. But I mean, it is almost magical. It's pretty fantastic.
Like that in nature?
I mean, a lot of crustaceans, like shrimp and crabs, they go through a metamorphosis like that as well. There are worms that look quite different. Actually, they don't look like worms as larvae, and then later they become worm-like. So yeah, there are other examples. Yeah.
So my question's related to the potential for dispersing across the sky. Do we have any idea about the dispersion of all types or anything like that for some of these insects that you found?
Right. Yeah, it's a good question. So the question was, how much potential for dispersal do some of these insects have to get from human beings? I mean, the answer is that there's a whole spectrum. From some of them, neither males nor females fly, and they might be very tiny, so they don't get around as much. Others are a little bit larger and can fly, so they have more capability there. In some, the males fly, the females don't, so then it depends if you're looking at maternal genes or paternal genes, what you're going to see. I showed one picture of a small wasp. It's a parasite, and it is flightless. And I actually thought we would see those as being very isolated, but they weren't. And I think what's happening there is that when they're parasites, their larvae are inside another insect that's probably flying from peak to peak. We looked at some spiders, and so young spiders, they let out a thread of silk, and then they can pick up on the wind, and they get blown around. So we saw a lot more of the sort of local enemies in the things that couldn't fly. But yeah, that was not the rule of thumb. Yeah.
What is some of the good about the bad? You did the good, bad, and ugly, but what's good about a mosquito besides that other things eat them?
What's good about mosquitoes? That's a tough one. Not all mosquitoes bite. Some mosquitoes are pollinators. So not all mosquitoes are bad.
Sure.
Obviously, eradicating mosquitoes because some of them are bad would be a bad idea. They are a critical part of the ecosystem. The larvae of mosquitoes are just in the water. They're not biting anybody. And yeah, they're such a crucial food source. So getting rid of them would be a terrible idea.
Sure. Yeah.
We just kind of got to deal with it.
Sorry.
I used to be
Anybody want to ask about ticks? A little bit of a harder sell there. All right. Anything else? Yeah.
So, invasives, for example, the germ spider, which I hate.
Yeah.
Are we seeing those from those peaks, and are they someone monitoring the impact that they have on these data?
They are not up that high, and I don't think they like the climate up that high, but They're certainly spreading, and I think that their abundance is bad for native insects. They are essentially setting up flight traps everywhere. I think it's good that mostly the Joros are becoming mature late in the year, and the majority of insects fly earlier in the year. So I think that limits the impact some. But they seem to be out-competing native spiders too. So we have lots of orb weavers that belong here, and they're not getting all the prey that they would normally get. So yeah, there's definitely some issues there. So kill them all you want. I got no problem with that. Yeah.
So, I study salamanders.
Okay.
So I really appreciate the inclusion of salamanders. I really appreciate just like the whole premise of we should be looking at life underfoot and the leaf litter. And I guess I have a question that's more general about insects and leaf litter beyond the Scott Islands and some of the Appalachians, like maybe more so like in the Piedmont.
Mm-hmm.
I'm sure you're familiar with like these invasive earthworms that are destroying our leaf litter and increasing the leaf litter competition. I was just curious whether you know of any research that's like a lot of that's being done in the Northeast. Do you have any knowledge of any that's being done like down here where there's a longer period where the earthworms are active? Our soils are generally already poorer as well.
I mean, I and other people in my lab have been doing collecting in Clemson forest where the earthworms in some places are very abundant. You definitely notice a difference, but we haven't really been looking at it very systematically. I do have a student right now who's looking at needle ants, and I'd be curious, do you know if needle ants are affecting salamanders at all?
So, I mean, I know they're a problem when you're sampling for them.
Right.
They draw it on you. I know, like in the coastal plain, there's some papers published about fire ants and some gopher frog species, some endangered frog species, as well as some endangered snake species that were in the nests there.
Yeah.
But I'm not super sure about needle ants.
Yeah, I mean, they're definitely eating a lot of the other arthropods in the leaf litter. So yeah, that's one we're looking at. But the earthworms is a good question. I don't know of anybody who's doing that right now.
Yeah, it just seemed like a gap in the literature for me. Like obviously that this affects salamanders as well, because salamanders use leaf litter.
I mean, honestly, even among entomologists, the leaf litter stuff's kind of a hard sell. A lot of people like to look at butterflies and big beetles and stuff like that. So I'm doing the same thing among entomologists. Like, you guys need to be looking at stuff in the leaf litter. So yeah, we need to draw more attention to that. I mean, I'd love to know more.
Yeah, for sure.
Yeah. So the needle ant is a species that it's actually been here for 30-some years, but they seem recently to be expanding. It's an invasive species from Asia, and they have a really nasty sting, and they eat all kinds of other insects.
Are they like fiery?
Well,
They have a big male.
they don't hound. They mostly nest in logs, so you do find them. I mean, we have them all over our backyard. But the thing about them is they go into native forests and are kind of fire ants mostly stay out in pastures and more open high areas. These things are going into the forest where there aren't many other economic concerns, and so they're just being allowed to spread.
Good insect.
Yes. So I'm concerned about them, but not many other people are.
Never heard of it.
Yeah.
Never heard of it.
Not aware of this or cases Okay, so there are slave making ants that go to ant colonies and steal those ant larvae. So yeah, you've got the story mostly. But yeah, those are here, but those are both native, and that interaction has been going on for millions of years. Yeah, that's amazing. Slave making ants are too lazy to raise their own young. herbicides, probably. Yeah. I mean, really, I don't think the insects are terribly susceptible to herbicides. My bigger concern is that just kind of driving down the road in the summer and spraying for mosquitoes. You know, there's just lots of other things that are susceptible to herbicides. So, I don't know. Ideally, I'd be very anti-chemical, but you know, you've got to like find the balance there. Yeah. I mean, again, I don't know if any comes with herbicides especially, but it's going to be killing other plants that certain insects like to eat. Yeah, they're pretty focused and narrow strip, so really not a huge impact. Yeah.
What do you know?
I don't know about fire ants and electricity.
They're always getting in electrical boxes.
I actually was going to put it out there, and then I thought, maybe you know, some people. Yeah. No. I mean, I did. I really thought about it. All right. I think we're going to wrap it up here, and thank you all so much for coming out. I really enjoyed the discussion, and yeah, I'm easy to find. Clemson's website. If anybody has other questions, they want to email me. Follow me on Blue Sky. I don't really tweet anymore, but yeah. Have a great night. I'm Halloween. What's up? I haven't given away a card in many years. Sorry.
You were talking about doing some dolly over here, the dolly? Yeah.
Oh, okay. Oh, like a computer nerd. Yeah, that's a good idea.
Except all over.
Yeah. No, that'd be really nice.
They're always looking for you.
Thanks. Yeah, that's a good idea. I mean, that would be great. I'd love to talk to you.